Hello, what's up everyone? My name is Pratiksha Pujari and today we are actually going to listen to the chapter Transpiration. So when we spoke about transpiration in absorption of roots, there was a gist which was given to you, a rough idea you already have about this. So just to put it again, when we talk about transpiration, we all know that whatever water absorption takes place in the parts of the plant, it all takes place through the roots. Okay. So when the water is actually being absorbed by the roots, it goes to all parts of the stem as well, which we call as the aerial parts. Okay. Whatever parts are in the air, we refer to them as the aerial parts of the plant, like the stem, the branches, the leaves, everything. Okay. The shoot, all those are aerial parts. So from the roots, it's all the water is going to the aerial parts of the plant. And then it is being utilized for various activities of the plant. Whatever metabolic activities have to take place, there it is going to be utilized. But only 2% of this is actually utilized, all right? Only 2% of water is utilized by the plant, okay, for its metabolic activities. What happens to the remaining water? The remaining one is going to be lost through transpiration, okay? Remaining is going to be lost through transpiration. Now, what exactly is transpiration? Transpiration is nothing but water loss through the surface of the leaves. Okay? Water loss through the surface of the leaves or some parts of the plant. Okay? It can be or some other parts of the plant. So whatever water loss is occurring from the external surface of the plant, it can be referred to as transpiration. Now, water loss is happening, water is actually going from the plant, okay, but transpiration is actually essential, okay. Transpiration is actually serving some purposes in the plant, okay. So what exactly are the uses? What purposes is transpiration serving, okay? So when we talk about purposes served by transpiration, the first thing we can talk about is, as I told you, those roots are actually pulling the water, okay? And when transpiration is happening, obviously it is happening because of solar energy, okay? So when the water is actually coming up, okay, so the roots are going to be pulling the water from down, and transpiration is actually happening through the leaves like this, okay? So there is a kind of water column which is created in the stem, okay? So there is a kind of a suction force, I can say, which is created in the stem, okay, and this suction force is going to help in water transport basically through all the parts of the plant, okay. Because of the absorption of the roots and because of transpiration which is happening, this water column is going to be formed, and that because of that suction force, there is going to be water transport which is occurring throughout the parts of the plant, okay. That is the first thing which is happening. The second thing which will happen in this particular case is cooling effect. Now, in the hot weather, in hot temperatures, okay, obviously the plant, we feel dehydrated in summer, right? Similarly, the plant is also a living being. It will also feel the need for water and it will feel dried up, okay? There will be drying which is happening. So, especially in the desert climate, especially in hot climatic conditions. So, whenever this water column is actually entering, whenever transpiration is happening, there is a suction force which is created, which is allowing the water to move along the stem. So, it is actually having a cooling effect, or it is kind of cooling the plant surface of the plant body, okay? So now if we have to measure transpiration, okay, that how much of transpiration has happened or how much of water loss has happened, how do we actually measure this, okay? So if we talk about measurement of transpiration, okay, there are a lot of methods to actually measure transpiration. Let's look at some of the methods. The first method is going to be called as the weighing method. Okay, it's a very simple method. Whenever you have to weigh something, what do? You actually weigh it with a weighing machine or scales, right? That is what you do. So this is what we are going to do in weighing method. Okay, but then let us think how exactly by determining the weight of the plant are we going to come to know that what is exactly the transpiration or what is the rate of loss of water which has happened in that particular instant. Okay, so let us look at that. So in this weighing method, what I'm going to do is I'm going to take a potted plant. Okay, so that potted plant is going to be covered properly, okay? So the root, every part of that potted plant is going to be covered from top to bottom so that there is going to be no water which is coming in or no water loss which is going to happen, okay? And then I'm going to weigh it before I do that. Before I actually do this, I'm going to weigh it, then I'm going to cover it and I'm going to keep it for like say few hours or 1 hour, whatever, as for a stipulated amount of time, for whatever time I want to measure the rate of transpiration or the rate of water loss. All right. So let's say, for example, I keep the potted plant for 2 hours. Okay. So after 2 hours, I'm going to take the weight again. And what I'm going to see is there is going to be a reduction in weight. Okay. So why has this weight reduction happened? Yes, the weight reduction has happened because of loss of water in this particular instant. Okay. So earlier there is water which is present, and later on when transpiration has happened, there is water which is lost from the surface of the plant, and there is no water around that the roots can actually absorb or can be lost through the roots. Okay. So this has plainly happened because of transpiration only. This can be done by this method also, or else what you can do is you can take something like like a test tube like this. Okay. Some kind of a test tube can be used. Okay. I'm going to use this test tube. Okay. I'm going to fill it up with water here. Okay. So the test tube is going to be filled up with water. This is another alternate method of weighing. All right. So in this particular test tube, I'm going to put some chute. All right. Some kind of chute is going to be put. Okay. So when I'm going to put the chute in this, I have to make sure that the chute is not having any roots. All right. So this chute specifically, I'm going to immerse in this water and it should not have any roots. Okay. And I'm going to keep this entire setup in a beaker, and I'm going to weigh this entire test tube. All right. So once I put it in a beaker, I'm going to weigh it. Again, after some time, I'm again going to take the weight. There is going to be water loss which is happening. So there is going to be a difference in the weight which we see. Okay. And also there is no absorption or water which can go from here, because this is only the shoot. There is no root. That is the whole purpose of not having the shoot with the root. All right. And whatever water loss is seen, that is equivalent to the amount of transpiration that has happened in that particular instant. Now talking about the second method of weighing the actual process of transpiration or the rate of loss of water which has happened is the potometer. Okay. Now when we talk about potometer, poto means drinking and meter means measurement. You all know that. So basically it is going to measure the water intake. Okay. So we are going to measure the water intake which is going to be equivalent to the transpiration which is going to be happening in this particular procedure. Okay. So there is an experimental setup here, as you can see. Okay. This is the setup for the potometer. All right. So this is going to be a potometer. Okay. So as you can see here, there is a beaker. Okay. There is going to be water which is filled throughout this beaker, and there is going to be a twig on one side. Okay. So there is a twig which is cut with a very sharp knife. So this is my twig. Okay. The twig is cut with a very sharp knife and it is immersed here. All right. Throughout the column, throughout the tubes, you can actually see water in this. This part of the tube which you can see here, this is colored water. And here it is just water. Okay. So I'm just written it as colored water so that you can differentiate between the two. Okay. So here you can see there is a graduated capillary tube. Okay. So this is going to be inside this is a graduated capillary tube, and there is a scale actually to measure it. All right. So here you can see there is a small space in between. As I told you, the entire column is going to be filled with water, but there is a small air bubble here, you can see. So this is going to be my air bubble. All right. So here we have my air bubble. Okay. So what I'm going to do is I'm going to put a twig here. Okay. And I'm going to set up the entire tube is this way. Okay. There is a horizontal graduated capillary tube. There is a beaker. There is colored water going to be filled in this. Okay. So the water is obviously going to rise by capillary action. Okay. Now, how do you actually make this air bubble is you actually bend the capillary tube, then keep it straight. So the water rises up by capillary action and bend it again so that there is an air bubble which is created here. Okay. And here you have this entire setup. Okay. Here you have the twig and here you have the reservoir water. I'll tell you why the reservoir is important. For now, just understand that this is the reservoir. And here you can see this arrangement. This is a stopcock, okay, to control the flow of water. All right. So this is a stopcock. Okay. It's like a tap. That's it. Anyways, so now coming to the actual experiment, how am I actually going to understand the rate of transpiration through this entire setup? Okay. So I've put this twig here. Okay. Obviously that entire air bubble is there, there is going to be a water column, okay. That twig is going to lose water through transpiration, yes. So transpiration is going to happen in this particular instance, right? So transpiration is going to happen. So when transpiration is going to happen, as I told you how it actually happens in plants, there is a suction which is created. It is going to be absorbed from the bottom, okay. So obviously if there is a suction which is created, the liquid will rise here and the air bubble will move forward as and when the suction is created. And depending on how much the air bubble is moved forward, you can actually measure this with the scale, which is with the graduated capillary tube. There is a scale to actually measure it, okay. And if you want to bring the air bubble back to the to its original position, what you're supposed to do it, you're just supposed to open the stopcock and adjust the reservoir of water so that the air bubble can go back to its original position and you can measure it once again, okay. So in this way, whatever water intake is there, how much ever the air bubble gets pushed or coming ahead, you can actually measure the amount of water intake by that particular plant, and thus by knowing the water intake, you can indirectly measure the rate of transpiration. Understood this? So this particular potometer we see here, there are various types of potometer. So technically this potometer setup which I've shown you here, this is called as the Ganong's potometer, okay. So there are various type of potometers, Darwin's potometer, a lot of types, Gary's potometer. But here this particular setup which I've shown you, it's for the Ganong's potometer, and Ganong's potometer is specifically actually estimating the water intake. So technically this potometer is not measuring the rate of transpiration, but it is actually measuring how much water is being taken by the plant. Okay, so that water is necessarily used for photosynthesis or whatever purposes, that can be estimated accordingly. Okay, so now this particular, there are some precautions which you need to take while actually setting up this entire experiment or with this entire potometer setup. Okay, so there are some things which you need to keep in mind. Okay, so note, all right, there are some things which you need to keep in mind while actually doing this entire setup. Okay, the first thing what you should keep in mind is this entire setup should be watertight. There's only one air bubble, nothing else. There has to be no other air bubbles present anywhere. So it has to be an ultimately watertight setup. Okay, so that is the first thing. Second thing what I'm talking about is the shoot, whatever shoot you're going to use here, it should be obliquely cut like this. Okay, so the tip should be something like this. It should not be like this. Okay, it should be oblique. Okay, so the tip should be obliquely cut. Why it has to be obliquely cut? Because that will actually provide more surface area. Okay, so more surface area is available for absorption. However, we must take care that there is no air bubble actually entering inside the twig. Okay, because if an air bubble enters inside the twig, the xylem, it will block the absorption of there will be no absorption inside the xylem vessels, because xylem is actually responsible for conduction of water, right? So these are some things which you need to take care of. All right. However, also there are some limitations. There are some things which are not under your control. For example, some of the limitations we see is, you know, you cannot keep the twig alive for a long time. So keeping the twig alive is a big challenge by itself. Okay. So keeping the twig alive. Okay. Secondly, this air bubble which you are introducing, it's not an easy task. It looks very easy. Okay, when you see the experimental setup, it looks very fancy. You can easily do it. You feel it. But once you actually start doing it, the introducing that air bubble is not easy. So introduction of air bubble, not easy. Okay, and the third thing, any kind of changes which happen in the temperature, okay, even the slightest changes which happen in the temperature is going to affect this entire setup. Okay, it can affect the rate of transpiration. So changes in temperature. So these are things which are absolutely not under your control. They are absolutely not something which you can take care of. Okay, so these are the limitations of using a Ganong's. Now let's see. Now as we've seen what the kinds of what actually transpiration is, let's see what are the kinds of transpiration which are actually there. Okay, so kinds of transpiration. The first kind of transpiration is stomatal transpiration. The second is cuticular transpiration. We'll be seeing each of these types in detail. Okay. And the third type we have is lenticular transpiration. Okay, so primarily there are three types: stomatal, cuticular and lenticular. Stomatal is actually through the surface of the leaf, which is through the stomata. Okay, stomata is basically where the exchange of gases between in plants actually takes place, okay, during photosynthesis or during respiration. Okay, so most of the transpiration which happens is a stomatal transpiration. Very little amount of water happens, water loss which happens either through the cuticle or the lenticle, which we will be covering later. Okay, so let's focus on stomatal transpiration first. It obviously happens through the stomata. Okay, there's a rough diagram of the stomata which I've actually represented here. Okay, so if you look at the typical structure of a stomata, okay, so this is stomata is for plural. There are stoma is, it's actually called as stoma. It's just a tiny minute opening which is present on the surface of the leaf. So this opening which you see here, this entire structure, this can be referred to as the stoma. Okay, and outside the stoma you will see two bean shaped cells here. Okay, these two bean shaped cells here, these, they are referred to as the guard cells. Okay, so depending on whenever there is a necessity for CO2, why does the plant take CO2? Whenever it has to conduct photosynthesis, or when it has to manufacture food in layman's terms. Okay, it has to take carbon dioxide inside so that it can manufacture food and then produce oxygen. So at that time these guard cells open up. Okay, so when these guard cells open up, that is the time actually transpiration is going to happen. Okay, transpiration as in loss of water is going to happen. So we will see how exactly that is going to happen in this particular case. So this way there are about 1000 to 10,000 of stomata present per centimeter square of the leaf. Okay, so now once the water is going to enter the okay, once it has to enter outside the stomata, or water loss has to happen, it is happening through the stomata. Transpiration is actually happening through the stomata. So now just for your reference, I've shown you the vertical section of a leaf. So this diagram which you see here is the vertical section of a leaf. Okay, so like every other structure, this particular leaf is also going to have various layers. Okay, so here this top layer of cells, there is a layer which is present on top. This thick layer which you see on top is nothing but it is the cuticle. Okay, so cuticle is a thick waxy covering which is present on the surface of the leaf. Okay, then you have something called as, here you have upper epidermis, and the bottom layer is nothing but the lower epidermis. Okay, so this is going to be called as lower epidermis. All right, so you have cuticle, you have upper epidermis, you have lower epidermis. Whatever is going to be present in between, okay, this entire structure which is going to be present in between, okay, this is going to be the mesophyll tissue. Whatever is going to be present in between these columnar, but the mesophyll is divided into two structures. The columnar cells which you see here, that is called as the palisade mesophyll. Okay, and these structures, these cuboidal cells which you see over here, is nothing but spongy mesophyll. Okay, all right, so when the water is actually covering, okay, it is coming through this, it is actually going to be diffusing through the mesophyll tissues. All right, so as I told you, diffusion always, as I've mentioned in absorption by roots chapter, diffusion always happens from higher concentration to lower concentration. So the water is actually going from the higher concentration areas of the pores of the leaf to the lower concentration area, that is the atmosphere. So here between the spongy mesophyll tissues, here the mesophyll tissue, there are a lot of intercellular spaces. The water is going to diffuse through these intercellular spaces. Okay, ultimately this water is going to form a thin film around all the spongy mesophyll layers. Here it is going to form a thin film, and ultimately it is going to diffuse into the substomatal region, and then this water vapor droplets, you can see these drops over here, this is nothing but the water vapor, these dots which you see over here. So these are water vapor which is actually coming out from here, from the higher concentration region to the lower concentration region. So as I mentioned, there is some kind of fluid which is present in the inside the vacuoles of the plant cell. Okay, that is called a cell sap. All right, so this cell sap, I've mentioned to you in absorption by roots chapter also, it's going to apply some pressure which is going to be called as Turgor pressure. Okay, so because of this Turgor pressure, there is going to be water which is going to be coming out of the cell wall. Okay, and it is going to be coming to the intercellular spaces. What is intercellular spaces? Spaces between two cells. Okay, so the water is going to get accumulated in the intercellular spaces, And ultimately, from the intercellular spaces, it is going to enter the substomatal region, that from the then to the mesophyll, and then from the mesophyll, it is going to enter into the substomatal region, and ultimately out from the substomatal region. When I say substomatal, it means below the stomata. Okay, and after the substomatal region, it will finally go to the stomata. Okay, it will be lost in the form of water vapor, and transpiration will finally happen in this particular process. Okay, so then there is more and more transpirational pull which happens as and when this process happens. I told you there is a water column which is formed in the cell. Okay, so there is a transpiration pull, more and more water is absorbed by the xylem vessels from the roots. Okay, and this transpirational pull can last as long as 50 meters. So trees as tall as 50 meters, this transpirational pull can happen. All right. And when we talk about transpiration, it happens more in case of dicot plants. Why? Because there are more number of stomatal openings which are present on the surface of dicot plants. Okay. So more stomata or more stomatal openings, present in case of dicot plants. So obviously, if there is more amount of stomata, what is going to happen? There is more amount of transpiration which is going to be happening, and ultimately there is going to be more amount of transpirational pull which is going to be happening in that particular case.